everybody. Today's class is about memory, which is a part of higher mental functions. So the three higher mental functions are learning, memory and speech. You came to know what is learning. It's a change in behavior of an animal based on previous experience and conditioned it without experience. Speech is expression of thought in the form of words, which was formulated and presented in the form of response to a spoken word, which had Broca's area, Wernicke's area. But today's class is about memory, which is registration, retention and recollection of information. These higher mental functions are complex. They are higher intellectual functions involving different parts of the brain functioning in a sequence. For all this, we need plasticity, cortical plasticity or synaptic plasticity, which involves growth of new dendrites, strength of synaptic connections, and synthesis of different proteins in response to internal and external stimuli. So, what is memory? Memory is the ability to store learned or acquired information and to recollect the same when it is needed. So it is retaining, retention and storage of information, or it involves storage and retrieval of information. It is needed for learning and integrating our experience into behavior. Bits of information are stored in the brain by changing the sensitivity of synaptic transmission between the neurons based on previous experience or activity. So, memory is the process by which knowledge of the world is encoded, stored, and later retrieved. This is the definition in the book called as Neurophysiology of Kendall. Or it is ability to recall past events at the conscious state or subconscious level. It is stored by changing the basic sensitivity of synaptic transmission between the neurons. And these newly established pathways or the facilitated pathways are called as memory traces, can be either activated or reproduce the memories. The different forms of memories are the explicit and the implicit memory. The explicit memory is declarative or recognition memory. It is a memory associated with awareness of or consciousness. For retention of this memory, hippocampus and medial temporal lobe of the brain are necessary. The two types of explicit memory. So, explicit memory is the process of registering and recollecting facts. It is further divided into semantic and episodic. Episodic memory is related to events. It is required to learn tasks such as riding the cycle. However, repeated training, memory becomes implicit. So, explicit becomes implicit. Whereas semantic memory is concerned with facts such as words, language, and rules. So, explicit can go on to become implicit with regular training or reinforcement, which can be positive reinforcement. So, the next major form of memory is implicit memory. It is also called as non-declarative, reflexive, or procedural memory. It is associated with acquired skills, habits which become unconscious and automatic. Here, hippocampus is not involved. So, there is no processing, it does not involve the processing in the hippocampus. The declarative memory or the explicit memory that are initially required for learning activities can become non-declarative once the task is thoroughly learned. So, once again, we have come to know the two major types, that is explicit and implicit. For one, we need hippocampus. And when the explicit memory is trained, it becomes implicit. So, what is the short term memory, long term memory, and intermediate memory? The short term memory is also called as memory, working memory. This type of memory lasts for few seconds to few hours. Hippocampus processes the information and converts it into long term memory. This conversion is consolidation. This short term memory is lost during damage to the brain and it is produced by continual neural activity. The signals travel repeatedly over the temporary memory trace. The molecular mechanism for short term memory is presynaptic facilitation or inhibition. The activated synapse releases neurotransmitters which can either facilitate or inhibit postsynaptic terminal for seconds to minutes. So, what is long term memory? This type of memory stores the information for years, from months to years, and it is stable and is not susceptible to loss in brain damage. Even in spite of brain injury, it's not lost. Long term memory is likely to produce structural changes at the synapse. So it's mainly the short term was chemical related, whereas the structural change can happen in long term memory, thus enhancing or reducing the conduction of signals. The structural changes noticed at the synapse are increase in the number of vesicle released sites or increase in the number of vesicles released, increase in the number of presynaptic terminals, change in the structure of dendritic terminals suitable to transmit long term. So, short term is converted to long term by the process of consolidation. Activation of short term memory repeatedly brings about structural, chemical, and physical changes required for long term memory. So, what is working memory? It is a form of short term memory. Here, the information is retained only for short term. What is it? It is retained for short periods. For example, to look up a telephone number before dialing. The information is retained for short duration when the decision has to be made based on the information. It gets terminated after the objective is achieved. Working memory has central executive and two rehearsal systems. So we will see in detail about implicit memory. Implicit memory is you get a lot of information to the brain. So that means the brain is fed with a lot of information on a regular basis with very little or no consequence is ignored. Only the important ones are stored. This results in inhibition of synaptic pathway. This effect of habituation, it is a negative memory. We have non-associated. That is, habituation is also called negative memory, where most of the information which are not important are ignored by the brain. Whereas important information associated with pain or pleasure is stored by enhancing the memory traces. So, what are the memory traces? Newly established pathway which can be activated and reproduce the memories or memory traces. So, sensitization is important information associated with pain or pleasure is stored by enhancing memory traces. This is a positive memory. It results from facilitation of synaptic pathway and the process is memory sensitization. What is associative? As you know, in learning we came across classical conditioning by Pavlov and operant conditioning, which is instrumental conditioning by Skinner. B.F. Skinner is called the father of. So, when you associate, for example, an unconditioned with a conditioned, there is pairing happening. So, I mean to say, if you ring the bell alone, there is no production of saliva is not produced. But before you keep the food and after keeping the food, you first ring the bell, keep the food and there is production. Over a period of time, only with ringing of the bell produces saliva. This is pairing an unconditioned and conditioned stimuli, which is classical conditioning. So this is associative, whereas operant is it is taught to perform a task to get a reward or punishment toward a punishment. Here the reinforcement and punishment are important tools to use to modify the behavior. Here positive reinforcement, negative reinforcement, positive punishment and negative punishment can happen. An implicit memory, as you all know, is associated with acquired skills and habits which become unconscious and automatic. And here the primary memory, priming of the memory, which is for recognition of words or objects. So we have come to know the mechanisms of habituation, which is negative memory, sensitization, and classical conditioning, and the main B.F. Skinner, who is the father of operant conditioning, wherein an animal performs a task to get a record or reward or avoid punishment. So habituation, as we saw, repeated application of neutral stimulus produces negative memory. So keep on actuated, there will be no response, it gets weakened. When a repeated stimulus coupled with unpleasant or pleasant stimulus once or more produces positive memory by enhancing the memory traces, by facilitating the pathway. Wherein you can see what happens here is in sensitization, the release of serotonin, the receptor 5-HT at a facilitator synapse. It binds to the receptor, activates adenyl cyclase, forms CAMP in the presynaptic terminal. The CAMP activates a protein kinase and phosphorylates a protein which blocks the potassium channel. The action potential is prolonged due to lack of potassium conductance. Prolonged action potential enhances calcium influx due to the opening of calcium channel for a longer duration. So its calcium is essential. The neurotransmitter release is increased due to excessive entry of calcium. Enhanced neurotransmitter release increases postsynaptic response. This is the mechanism of sensitization, which is positive memory, and the molecular basis has been explained, which is based on the decreasing the potassium conductance and increasing the calcium conductance, thereby enhancing the neurotransmitter release, which increases the postsynaptic response. Classical conditioning, we saw that somatic, visceral and other can be paired, and or otherwise a large number of somatic, visceral and other neural changes can be made to occur as condition. The conditioning of visceral response is called as biofeedback. The changes that can be produced include alteration in bowel movements, heart rate, blood pressure. Conditioned decrease in BP has been used for treatment of hypertension, which is a classical example of biofeedback. Or it can have positive and negative reinforcement, which is about the classical condition which is associative and non-associative. Whereas in operant conditioning, you saw there is condition avoidance reflex. It is a motor response to an animal to avoid an unpleasant sensation. It is a form of negative reinforcement. In this reflex, learning happens by operant conditioning. So what's the molecular basis of memory? Memory is dependent on the alteration in synaptic connections involving protein synthesis or activation of the related genes. So either there is alteration in protein synthesis or activation of genes. As you know, the conversion of short-term to long-term, which is called consolidation, is affected by shock, anesthesia, concussion, resulting in retrograde amnesia. So what happens here is the molecular basis is the RNA content in the neuron is changed, newly formed mRNA is moved to axons. There will be a change in the shape of dendritic spines. The number and size of the presynaptic terminals will increase. More neurotransmitters will be released from the presynaptic terminal. There will be formation of new neurons and the long-term potentiation and depression. For long-term potentiation has a role in establishing memory. There are two phases, the early phase and the late. Early is involved in short-term memory and the later is which has AMPA receptors and the NMDA which is blocked by the magnesium ion. Once the sodium conductance increases, the magnesium is expelled, extrusion, the calcium enters and the long-term potentiation occurs. That is the basic principle of long-term potentiation which is involved. Or if it is weakened, most of the time the information is ignored, means if there is no information of that nature, it is weakened and the synaptic connections are lost. So long-term potentiation has a role in establishing memory. The NMDA, that is N-methyl D-aspartate receptor, will act as a calcium channel and mediate long-term potentiation. The NMDA receptors are activated by voltage and chemical signals. So the different parts, that is the cerebral cortex, the thalamus, the hippocampus or the limbic cortex are involved. That is the hippocampus, the parahippocampal area of medial temporal cortex, the prefrontal cortex is concerned with different aspects of memory. So we will see in detail about this. In habituation, there is gradual inactivation of calcium channels which results in negative memory. Whereas in sensitization, when we pair it with a noxious, which can be of pain and pleasure, discharge of serotonergic neurons on the presynaptic terminal, causing short-term facilitation due to calcium-induced change in CAMP levels. Long-term changes involves potentiation of protein synthesis and growth of pre- and postsynaptic neurons, resulting in a greater postsynaptic response. In long-term potentiation, there is increase in postsynaptic neuron calcium. It happens around the hippocampus. The mossy fiber is LTP, which is NMDA independent, whereas the Schaffer collateral LTP is NMDA receptor dependent. So LTP has two phases, mainly because of the AMPA receptor, AMPA, and NMDA receptor. What is NMDA again? N-methyl D-aspartate, which increases when which is blocked with a weaker signal. Only when a stronger signal is given, NMDA receptor is open, which is blocked by the magnesium. With the conductance entering through the AMPA receptors, calcium, sodium enters, and increased action potential conductance causes magnesium to expel and calcium increases, causing long-term potentiation in the hippocampus. In long-term depression, hippocampus is another part that is produced by store stimulation of presynaptic neurons. In cerebellum, phosphorylation of glutamate receptor 2 subunit of AMPA receptor happens. So encoding of receptor. So during the process of consolidation, bits of information are stored and grouped into similar types. This process is codification. It helps in effective storage and easy retrieval of information. It happens in basal ganglia cerebellum, also the vestibular ocular reflex, eye blink reflex, as we discussed. Rehearsal helps in what is called as reverberating circuits. So consolidation of memory and its long-term storage depends on repeating and rehearsing the information which is gathered. Rehearsing enhances memory based on reverberating circuits. Whereas when you encode an explicit memory, It involves the central executive system, which is present in the it feeds the information to rehearsal system. The rehearsal systems are the verbal system for retention of verbal memory and the visuospatial system for retaining visual and spatial aspects of object. As we saw, working memory is nothing but form of short-term memory. It is retained for a short duration when the decision has to be made based on information. Like opening the telephone book and remembering the number only for a short while. Hippocampus and medial. You can see how it is connected with working memory and destruction of ventral hippocampus. There is loss of short-term memory, so hippocampus is essential for short-term memory. As you all see, the parts of and can form new long-term memory. This is mainly due to CA1 neuronal destruction. So CA1, CA3, which happens in dentate gyrus, which is in the parahippocampal. So unimodal and polymodal association areas, mainly the parahippocampal and perineal, there is a crossover. The mossy fibers as the hippocampus as CA1 and CA3, and collateral and mossy fiber, which we saw in long-term potentiation. The process of explicit memory involves encoding, consolidation, storage and retrieval. So as we saw, what is encoding again? It is during the process of consolidation, bits of information are grouped and sorted out. It helps in effective storage and easy retrieval. So what is memory consolidation? The cerebral cortex gets flooded with information. It is processed and about 5% of information is transferred to short-term memory. Short-term memory serves as temporary storage for the data. Necessary information is transferred to long-term memory under the influence of the following factors. So, rehearsal, association and emotion. fMRI, functional MRI and studies, the word recall takes place in the left frontal and the parahippocampal cortex. The visual recall is in the right frontal and bilateral parahippocampal. In alcoholic, there is a pathology in mammillary bodies of the hypothalamus. Amygdala is concerned with emotional memory. What is confabulation? Some people lie honestly. It is due to lesions in the ventromedial portions of the frontal lobe. So before we go into this, we need to know what is memory consolidation. So as you know, the cerebral cortex gets flooded with a lot of information, but only 5% of the information is transferred to short-term memory. The necessary information is transferred to long-term under the influence of following. So this is consolidation. This is mainly based on rehearsal, association and emotion. In rehearsal, because of reverberating circuits, consolidation of memory and long-term storage depends on repeated and rehearsing information which is gathered, which enhances the memory. First one. See, facts can be associated, as we also explicit and implicit, what we are saying. Facts can be remembered better by tagging and associated with newly acquired information with already stored information in long-term memory. Tagging rule-related events or information with incident will improve the memory. Learning and in an emotion, learning enhanced memory will be better in alert, motivated, aroused state. As you all know, norepinephrine is the neurotransmitter involved in memory consolidation. Release of norepinephrine enhanced during stress level. Increased levels of norepinephrine improve memory. Memory consolidation involves transfer of information from long-term memory. It involves merging of new information with already existing category stored in the information stored in the cerebral cortex. So the way in which hippocampus is involved in explicit memory, and which has central executives and two rehearsal system, and it involves encoding, consolidation, storage and retrieval, and these are the areas concerned. So loss of memory can be either short term and long term, which is amnesia, which can be retrograde and antegrade. So amnesia is loss of memory due to brain damage, psychological trauma and other systemic diseases. Sedative and hypnotic drugs can cause temporary amnesia. What is retrograde amnesia? Retrograde is the inability to recall recent memory, recall events. So there is loss of existing and previously formed memory. For example, old memory from childhood is unaffected. Dementia results in gradual retrograde amnesia. It occurs during injury to the brain. Whereas anterograde is a condition in which subject is unable to establish new short-term and long-term. He is unable to establish new short-term. It is due to damage in the hippocampus. Hippocampus is required for formation of new memory. Both together is global. When it is related to senses, it is agnosia. So dementia can be two types, Alzheimer's and senile dementia. It occurs in middle age due to atrophy of hippocampus and entorhinal cortex. So what is Alzheimer's disease? It's a condition with progressive loss of short-term memory followed by loss of other functions of the brain. So the pathophysiology is loss of the neurochemical disturbance are loss of cholinergic projections from nucleus basilis of Maynard to hippocampus, amygdala and all parts. The hallmark, the cytopathology, the pathognomonic finding is neurofibrillary tangles along with senile plaques. These are aggregation of cells and degenerated fibers around beta amyloid peptide core. The damage is caused by beta amyloid, which is mediated through glutamate and NMDA receptors. Damage is due to increased influx of calcium ions into the. So what are the features? There will be loss of recent memory, lack of spontaneous activity, loss of intellectual functions and spatial orientation. So the most of the time it's the atrophy of the hippocampus and entorhinal cortex. It's common in middle age. It is due to deficiency of acetylcholine and structural change in brain areas involved in cognition and memory. So what is intercortical transfer of learning? As we see in memory, learning together, unless until there is learning, whatever you learned has to be stored and retrieved is the memory. There are two studies, the split brain and the corpus callosum. If animals are conditioned to respond to a visual stimulus with one eye covered, and tested with the blind eye folded to the other, it performs conditioned response. This is true even if optic chiasm has been cut, making the visual input from. So this is when the anterior and posterior commissure along with corpus callosum is sectioned, that is split brain. No transfer of learning occurs. This demonstrates that neural coding necessary for learning and memory has been transferred somehow to the opposite cortex via the commissures.